If we have a gene sequence but do not yet know its function, let's discover its role step by step. We begin with bioinformatics analysis. First, we compare the sequence with genes and proteins whose functions are already known. Using sequence alignment and similarity searches, we can identify conserved domains, functional domains, and related genes across species. If our gene resembles a known enzyme, transporter, or transcription factor, for example, this gives an initial hypothesis about its possible function. But this would be only a prediction. Sequence similarity can suggest a function, it does not establish it. Now, we need to test this hypothesis experimentally. So we move to gene expression analysis. We can measure the gene's expression across different tissues, developmental stages, or environmental conditions using approaches such as qPCR or RNA-seq. When needed, we can also determine where the transcript accumulates within the tissues. For example, if a gene is strongly expressed in the roots, its expression increases during drought. This suggests that it may be involved in the plant's drought response. Now, gene expression tells us when and where a gene is active, yet it does not establish what the gene actually does. To test the functional contribution, we can use loss of function analysis. The goal is to determine what changes when the gene's activity is reduced or eliminated. This can be achieved by disrupting the gene or reducing its expression through approaches such as knockout or knockdown. We compare the modified organism with the control and examine the resulting phenotype. For example, if plants with the disrupted gene becomes more sensitive to drought, while control plants remain more tolerant, this provides evidence that the gene contributes to drought tolerance. Loss of function therefore asks, what happens when the gene is no longer active? We can then approach this question from the opposite direction using the overexpression analysis. Here, we increase the gene's activity and examine whether the phenotype changes. If increasing the gene activity enhances drought tolerance while reducing it decreases it, then these complementary results provide stronger evidence that the gene contributes to drought tolerance. At this point, we move to validation experiments. The researchers can also investigate where the gene product acts, which proteins or genes it interacts with, and which signaling pathways it may influence. Importantly, scientists do not necessarily perform every one of these experiments for every gene. This is not a fixed checklist. It's a process of building and testing hypotheses. Sequence analysis generates functional hypotheses. Expression analysis places the gene in a biological context. Loss of function and overexpression experiments test its contribution to the phenotype. Validation experiments strengthen the causal link, and molecular studies help explain the underlying mechanism. By combining these complementary lines of evidence, scientists can move from an unknown gene sequence to a supported model of its biological function.